Okay, so what I'm going to talk about today is reducing the cognitive footprint of brain surgery, and I'll kind of get into depth on ideas on how do we have more functional patients. And this is going to be quite different than the previous talk, but it has some things in common in the sense that it's a big data-based approach. So what we're trying to do is take the patient on the left as surgeons and make it look closer to the patient on the right. And that means put a hole where the tumor is, remove as much tumor as we possibly can, and to do it with the least amount of problems. Now, when it comes to things like language or motor function, we're not bad at that. We actually do pretty well with some of the techniques we have. But one of the challenges as we began to really think about this in a more critical way is that we can obviously always improve. And anybody who spent a lot of time talking to these patients knows that there are a lot of people who move their arm and leg fine, they talk reasonably okay, but they're not really normal. And again, I really would like to point out that as we begin to think about what's the next phase, that we can hold ourselves to a higher standard than moving all four and talking. So whether we admit it or not, there is anatomy there. There are brain networks and functions happening. And when we talk about non-eloquent brain, it is really kind of we really judge this as a very oversimplification of one of the most complicated systems in the world, or in the known universe, to be honest. The brain didn't evolve the frontal pole as a CSF sponge. It's doing something. And even if we don't know what it's doing, it's still there. It is still doing it. So the idea that there's this non-eloquent part of the brain where the risk is minimal and we can't hurt the patient, so you can pretty much do whatever you want, really is a outdated, multi-decade old concept that really underlies the fact that people didn't have any real clue how the cognitive system worked, and that there's some redundancy built into that system that allows us to get away with things, but we don't always. And again, there's a lot more of a cognitive decline in these patients than we really admit, and that this has consequences. We know that patients, as their cognition goes down, become less interested in having additional therapies like CAR T cells. So when we start thinking about this, there's a lot of people who've said that, well, awake surgery did very well with language and motor. Maybe we can use this for higher functions. And there's various techniques that various people have put out there, but I've always been skeptical of them because, in part, it really is hard for me to grasp the idea that we can look at a white blob with lots of different microscopic anatomies running through this with our naked eye, and take a stimulator and stimulate around that and understand the concept of multi-network redundancy. And maybe some of these functions really aren't localized to a place, or maybe it's their collateral paths. And this is something that human beings are just not very good at grasping with their brain. But more importantly, some higher functions are just things that are not easy to test in the operating room, no matter how creative you are or how cooperative a patient is. And of course, if you think that you can get a 68-year-old GBM patient to do with a lot of swelling and edema and some aphasia to do higher level cognitive tests, I think you're kidding yourself. And ultimately, this really causes me to think that we need a better way to think about how to do this if we really want to make a difference and impact and leave our patients as functional as possible. Now, one of the things about it is that when we start thinking about things like the onco-functional balance, DTI and fMRI have been around for a very long time. We've been talking about them for a very long time, and they're not new, and there's not really a lot of controversy that they provide some insight in how the brain works with some limitations and caveats. There are over 550,000 papers last time I looked at this. One of the things that I will try to convince everyone on is that it isn't that these things are the ground truth, the only answer to the question. But what they do is they begin to shine a little light in the black box and help us make reasonable guesses and inferences based on data. Whereas right now we've used this based on gut instinct or hope, which neither one of which are a very good technique for cognitive preservation. And so one of the questions is, given that this has been around for a very long time and everyone knows about it who is doing surgery at least, and there's not a lot of controversy, or are these techniques real and give you some kind of insight? Why aren't we doing this on everyone who walks in the door? Why aren't we doing this post-operatively? Why aren't we checking patients and really looking in and leveraging the massive amount of research that's on here? And it's really simple, in my opinion, having actually tried to do this at an institution where we didn't have PhD experts. The coding is painful, the software is not very good, and requires a lot of time to string these pipelines together. And anytime you put in the workflow of a normal patient flow a PhD, forcing them to do a lot of the work, they have other things to do. And this means that this only happens at special times when there's plenty of lead time to get this done. And this means we don't get it on really sick patients. We don't get it on glioblastomas. We don't get it on people whose surgery gets scheduled in a short time period for whatever reason. And what this means is that we don't do it enough to really know what we're doing with the information. Forget the fact that the brain is the most complicated thing in the known universe, and we need things like artificial intelligence to help it make sense. So what we get are these pictures that look like this. They're pretty, they look good in grants, but we don't really know everything that this is really telling us. What are all these fibers doing? If you're smart, you probably know that those red fibers coming out at you somewhat somewhere in there is the language system. But other than that, what are all the rest of them doing? Who are the guys involved in cognition? And the fact is that this is an extremely high dimensional problem. It's a million dimensions that need to be taken into at least. And human beings are just not good at solving million dimensional problems. We struggle around four. And so we need things that can cut the number of dimensions down to a tractable, understandable, and learnable paradigm. And in this regard, techniques like parcellation, which is what you're seeing here, provide a very rational way to do this. And parcellation basically just means dividing up the neocortex into areas based on some kind of criteria. So everyone probably who has had a basic neuroscience knows at least one parcellation scheme. It's Brodmann's, where he did it by bicidal architecture. What I'm showing you here is the Human Connectome Project one that was published in Nature in 2016, where they did this using a variety of different imaging modalities, mostly fMRI based. And what they basically showed was that there were roughly 180 cortical areas. And when this came out, almost as soon as it came out, I immediately went to work on this because I realized a lot of things were really important about it. Most notably, that the brain really could care less what we call a gyrus. It tucked areas under the opercular cortex, it tucks areas into the sulcus. It doesn't really care that these are inconvenient units of understanding for us. It didn't take that into consideration. And so for order for us to really make progress, we need to talk about the brain using a nomenclature that better fits its organization. And more importantly, in order for us to learn what we're doing wrong, we need to be able to communicate and say, well, you shouldn't cut out area 55B because it does this. And those kind of things are essential. This is what was essential for taking inoperable skull-based tumors and making them operable, was so we could discuss and communicate results. Well, this gives us a framework to do this. And so I began doing what the HCP was really not aimed at doing, which is getting it back to just more conventional anatomy, which is we mapped out every one of these areas, what it connected to, what ran through different white matter bundles. And when this got really out of hand, thankfully, Nelson Ishiki let me put this in an issue all together in one place. But one of the challenges that we had was it's just a huge amount of information. And in order to really leverage it in the real world, it's not realistic to ask everyone to memorize 500 pages of anatomy, a lot of which looks very similar to itself. And so you have to be able to atlas the brain, which means that you take a list of where these parcellations live and register it onto the brain that you're talking about. And so standard techniques have involved affine warping or different forms, or surface matching or things like this. And then you can see how that looks. It does okay, but of course, when you assume that a place in space is Broca's area, then you're going to be off sometimes because people have different sulcation patterns, and you're going to be really off when this is the problem. So we know that not only is this brain distorted, this patient may have had surgery, and a lot of the software doesn't do very well with parcellating this. And that was what I found to my horror when I really wanted to take this into the real world. So I got together with people who could actually tackle this a different way. And what we did was we said, let's do this based on connections, because if the tumor shifts the brain, the connections are going to shift too. And so what we did was we taught a machine learning tool how to recognize a voxel of every part of the brain. So, for example, in this case, Broca's area, based on who it's connected to. So it knows that that is part of Broca's area because it talks to the Wernicke's areas. And this is not part of area 44 because it doesn't. And it goes around and does this for every voxel in the brain and makes an assignment based on the wiring diagram. And what this means is it's much more robust to individual variability because you can't put a voxel of a parcellation in a sulcus because there are no tracks. More importantly, if it tries to put it in the middle of a GBM necrosis region, there are no tracks there, so it won't map anything. And what allows this to happen is that if there are displacement or even potentially some degree of rewiring, it can at least say that these are the best candidates for the people who have the connections of Broca's area. And this has a really significant benefit in brain tumor patients. So what that does is that if you look at a case like this where we didn't even operate after removing the frontal lobe, this GBM came back with a vengeance. But one of my residents dutifully ordered a DTI scan, so I put it in there and saw what it did. And what you can see is that even though the insula on the right side or the left side of the brain, right side of the scan, is shifted pretty anteriorly into the cavity, it still is able to handle where the basal ganglia is. It's able to tell us what areas aren't there anymore, and to really handle the fact that the brain is extremely abnormal. And so what that means is that we can, once we get an atlas registered, because we've done so much anatomic work, we can leverage that and to make reasonable inferences about what the brain is doing. So we know this tumor is bad, and we know that if you resect it, you often can get some pretty significant cognitive blunting. Many times these patients come in with cognitive blunting of memory. And that never made sense to me as a resident why this was a bad brain tumor to operate on, because we got the splenium all the time. So why is that part of the corpus callosum magically in a tumor case? Difficult. But now we can actually see it. So we can basically pull out and create models of things like the language network and sensory motor network. We can see that this tumor, particularly the parietal butterflying portion of it on the left side, is tucked into these networks pretty intimately. But even worse, we can actually see what the real problem is and why these patients have cognitive problems. And that's that the default mode network, which is the central cognitive network of the brain, or one of them, is almost 180 by this tumor. That's the fundamental nature, because this tumor sits, because it's following the corpus callosum, it's inherently going to be wrapping around the cingulum where that network is located. And so not only do we know that it's bad, but we can begin to start thinking about, well, maybe we can get at least some of this out, because we know that just radiating this is essentially not much better than sending the patient home on hospice. These patients do very badly in my experience. And so what we can start thinking about is, can we do something that is not that bad, and to at least attempt to go at this type of tumor? And again, this is something that we would have never had an idea of how to tackle this thing in a way that wasn't completely crazy. So while it may not make that tumor a totally operative tumor, what I really want to highlight is that, but when we have better understanding of what the problem is, we make better decisions. So one of the things when we began to look at the connectome is, if we're able to map this, and we know that higher level brain functions result from large scale networks of areas that are not necessarily directly connected, and their intercommunications and their functional connections, then it's important for us to try to tie this down to exact anatomy, because there's a massive literature on where things are, what parts of the brain are activated in various tasks. And you could actually use a technique called meta-analysis to take that literature, take all the points that have ever been cited in a study, and make it into a three-dimensional Gaussian. That then you can identify who lies in that Gaussian and make a map of a network. In this case, this is the speech network. And as we began to do this, there's some things that we have learned that I didn't know 10 years ago, and that we can begin to tap into a vast corpus of understanding of the advances in neuroscience that have happened over the last 30 years, and begin to think about the brain in a more modern way to make better decisions. So to kind of highlight a couple things that we've learned, one of the things that's come out of other people's work especially, but also our ability to tie this into anatomy, is the concept of control networks. And so control networks, you may know them as the quote association cortex. That's obviously in modern terms not really a defendable term. But what the control networks are is they have central core areas that oftentimes define the behavior of other areas. So for example, the language system, it lies in the neighborhood of the default mode network, and the default mode network, or the DMN, is really drives how the language system works. And so oftentimes the way to think about them is that it alternates with an alternate network called the central executive network, which is a more goal-driven external network. And they work together by synchronizing their behavior, actually asynchronizing their behavior, to determine how do we allocate resources, where are we putting our effort. And in fact, actually, the inability of this system to switch correctly is actually the fundamental basis of many aspects of mental illnesses. In fact, most mental illnesses have some problem with the system. There's a third network called the Salience network, which is more discovered recently, which is actually probably equally important. And this network is actually involved in the switching. Now, once we begin to tie this down to exact anatomy, we can make maps like this, where you can actually see where in the brain this network is located. The DMN is really a dominant cognitive network and is involved in internal mental thought. You can see it has three components: anterior cingulate, the posterior cingulate, and the lateral parietal area. The salience network is also known as the cingulo-opercular network, and is a middle cingulate and anterior insula network. And the central executive network, at its core, has a frontal polar, an area called 8C, which is an extremely highly connected part of the dorsal or prefrontal cortex, and the supramarginal gyrus. And again, you can see that the intense, large span that these networks have been tied down to in numerous studies. And really, this highlights that we haven't really been thinking about these networks very much. But at the very least, it seems worthwhile that a first step towards minimizing our cognitive footprint is to not cut through the center of these if at all possible. That doesn't mean we can always save them or should save them. So if there's a GBM growing into the lateral frontal pole, I don't recommend leaving this thing growing, because as a method for saving function, we know that if you don't control the tumor, the function is gone. But what this means is that we shouldn't do it on accident. We should try to, whatever possible, to minimize the damage we do to these networks, because over time, this is likely to increase the cognitive footprint. And it also begins to teach us some things that we know intuitively, but never had a detailed understanding of exactly how it happened. So we know that medial frontal injury is bad and causes things like abelian and kinetic mutism, but now we can actually look at how that network switching is happening. And we can actually see that there's this little strip where the DMN and salience meet the supplementary motor area. In fact, the supplementary motor area is made up of parts of the DMN and salience wrapping around a motor area. So a lot of it has a cognitive component. It makes sense, because you have to make a decision to initiate movement. But when we start looking at this and we start comparing it to lesion studies like this one from Harvard, it shows that in order to get akinetic mutism, you have to really destroy that area or the thalamus. So it shows how important this really is. And also, when we talk about someone being quote unquote bifrontal, what we really mean is by this. This is the injury site. And so we haven't really spent a lot of time thinking about how to preserve this, but I would argue that, given that we move heaven and earth to avoid arm weakness, which is a much less devastating problem than leaving someone a zombie, in my personal opinion, then I think that we really need to think about where we put our priorities and our oncological decision making. So how do we actually do that? And science is interesting, but one of the thoughts we had when we were writing this book is, how do we basically determine safe cuts in the brain based on what we know? And so in that, you can define glioma surgery as a series of disconnections, because fundamentally, even if you start in the tumor and work out circumferentially with the CUSA, at some point you're making a division between what's staying and what's going. And whether or not you're aware of it, you may or may not be cutting next to these networks. So it's best if we think about how do we avoid cutting them. And so we came up with some ideas on how to minimize that damage. And actually, we've automated that into a workflow where you can essentially just pick what part of the brain you're operating in, and it will generate a set of objects of what's at risk. And this could be network models or tracks. And so when we start looking at things like thinking about the glioma surgery as fundamentally a disconnection, we look at a tumor like this differently. So instead of just going in there and saying, I'm just going to suck until I get to the ventricle, we actually think about it in terms like this. where we make a decision on how do we make cuts to minimize damage to networks. Now, that cut into the with the DMN, you may choose based on the grade of the tumor, the clinical exam of the patient, et cetera, that this is just not either worth saving or possible to save. And you may just accept that problem or accept that it's inevitable. That's a decision, that's a clinical decision. But one of the things is that at least here you're making it on data. And you're not going to accidentally do this and then be surprised that the patient's doing worse than you thought they are. And so, again, if you look at it in this regard, you can still do fairly aggressive resections and minimizing unnecessary collateral damage. Again, you see there we left tumor in the basal ganglia. That's not an accident. That was done on purpose for obvious reasons. When we think about reducing the cognitive footprint further, we have to acknowledge that cutting to the core of the networks is probably really important, and probably we're doing a lot more damage than average by destroying the default mode network wholesale, that this may still not be enough to completely address this issue. Because sometimes higher level functions involve mixture of networks. For example, the math network involves multiple parts of different networks, including the dorsal attention, semantic system, parts of the Salience network, and the white matter that connects them. And so other techniques for beginning to think about what are we doing wrong in patients that we make worse involve looking at the brain more like a global network and utilizing really sophisticated techniques from network theory and what's called graph theory to try to look at hypotheses on the brain. And we've done a lot of work on this, but I find that it's easiest to start with the simplest and most low-hanging fruit in a talk of this length. So if you look at this, how do you make the brain into a mathematical equation? So let's say you take a whole brain DTI, you parcellate it, and then you make it into an equation where everything is the parcellations are those dots and the lines between them represent how many fibers go between that. And now you've basically abstracted away the anatomy and just set this into a large mathematical equation called a graph. Well, you can do experiments on it. And one experiment you can do is you can ask who are the hubs of that network, how does information flow, and who is the most connected area. And there are different ways to measure it. The one on the right is called page rank centrality. It's actually Google's initial intellectual property was using this as a search algorithm. They've obviously become a lot more sophisticated than this. But you can do this in the brain where you say who is the most connected part of the brain or the hub. And it turns out that hubs, without any question now, have different genomics, use energy differently, and are differently susceptible to disease. In fact, Alzheimer's disease is thought to be a hubopathy. Now, whether it's cause or effect, it's hard to say, whether the disease targets the hubs or you can't get symptoms of the disease without having a hubopathy is, a very complicated question. But the bottom line is we know that Alzheimer's disease has loss of hubness as one of its fundamental characteristics. So one of the things about these kind of things is that it's easy to get off into deep science, but when we start talking about practical day-to-day applications, it's important to translate this back into stuff that we know. So when we talk about the term eloquence, what do we mean? We know that it's an area of the brain that if you cut into it, you get a deficit. But what does this mean from a connectomic standpoint? And so we looked at this and we just said, who are the biggest hubs in the brain? And this is the top 10. And if you were grading an AVM or asking neurosurgeons, is this eloquent? These guys would get an obvious yes. So you see there's the motor cortex, there's parts of Broca's area here, primary visual cortex, primary sensory cortex. And these guys probably should be on that list, but again, they have more abstract concepts, and it take me a lot longer to explain what the deficit that you get when you cut into these areas. And so hubness and eloquence are a lot of the same thing. We get deficits when we cut into hubs because they're very connected and they're very important. A lot of times they're very connected because they're connected to the outside world, but whatever it is, hubs are important. And we know that from numerous other ways to prove this. So the next question is, what if someone has a hub in an unusual place that's unique to them? Well, it seems reasonable to ask the question, maybe these are the patients that when we do a surgery, they don't do as well as other people getting the same surgery. So we know that there's patients that you remove a tiny glioma from, and they never go back to work, and they have depression or something else. And is it possible that what we did was we just took a hub out and didn't know that it was a hub? So we asked that question, and this is a paper in Journal Oncology a few months ago. And basically what it shows is that the answer to that question is about 8%. About 8% of people have a hub where nobody else has a hub. And so if you look, it's those guys at the very bottom, those are parts like the frontal pole and the medial parietal lobe that we don't think of as eloquent. But whether this tracks against clinical findings that we care about is another question. It's a much more difficult question to answer that we're in the process of trying to answer. But the bottom line is it raises the interesting point that we might be able to actually utilize these kind of techniques to figure out where the traps are and maybe make different decisions, particularly if we're talking about supramaximal resections and things like this. Finally, Connectomics has the ability for us to actually look at the connectome and potentially change it, because there are FDA-approved treatments like transcranial magnetic stimulation, which is approved for depression and OCD in the United States, but it can be used off-label for other things. And we've been using it for brain tumor patients for rehabbing them for many years now. And even though that was supposed to be a no-no because of the seizure risk, we've never had a seizure with the techniques we use, at least, and we've never had anybody get worse. And in many cases, we've seen recovery trajectories that are far faster than we would expect. So while these patients may have recovered on their own, it certainly has a huge neuro-oncological benefit if you take a patient who has SMA syndrome and instead of waiting 6 months, you get them better in 4 days, as far as getting them into additional treatment and minimizing complications. So this is what the device looks like. It sends a magnetic pulse into the brain. It makes a field like you see on the right. And one of the questions when we start talking about brain tumor patients is because of the extreme complexity of their connectomes, and this extreme complexity of the nature of the deficit, it really involves using bigger scale machine learning type techniques to even figure out where the problem is. Because what we found is that many patients have intact cortical spinal tracts, but weakness. And when you look at these people, what you find is that there are abnormal functional connections, meaning that the bold signals are out of sync compared to normal individuals inside of the motor planning areas or between the motor planning areas and the basal ganglia, things that would actually explain it. And that this is actually extremely amenable to desynchrony or resynchronization with TMS. And so again, you can't re-anastomose tracts that have been cut. So if you cut the corticospinal tract, there's not a lot you can do, but that's usually not what's happening in our practice. So when people have temporary or even potentially long-term weakness. And so what we can do is look at the functional connectivity. And for those who are not familiar with functional connectivity, it's the idea that during the resting state, different areas are supposed to be firing together or not firing together based on a lot of things, particularly their direct physical structural connections, but also the pulsatile rhythms of the basal ganglia. And this is the underlying aspects of brain networks. But in disease states, particularly things like mental illness, there's an enormous corpus of data showing that sometimes things start firing together that are not supposed to fire together. And so what you can do is you can actually compare the bold signal correlation between every area and every other area. And using things like machine learning, you can determine, is this an outlier? So for example, let's say, for example, these areas are not supposed to synchronize. You could actually make a comparison or find that there's under or overactivity. And this can help drive where you decide to aim the magnet to, again, augment rehabilitation. Again, like I pointed out, using things like functional connectivity, there's been enormous amount of work. This is a paper in Nature Medicine. And what they're showing is that mental illness is an extremely heterogeneous disease, and functional connectivity is a very good way to sort out the heterogeneity, and often finding that these are actually multiple diseases that are very similar to each other, but are fundamentally different. So when we start looking at how do you compare 379 areas with each other, you get a massive graph like this. So if you look on the X and Y axis, these are all the regions of the brain, subcortical, cortical, et cetera. And the Y axis is the same. These are just comparisons of everyone with everyone. The red dots here are areas that are correlated with each other, meaning they're firing together more often than normal. And then this is the areas that are firing anti-correlated, they're opposite. So one's firing, the other one's not. But one of the nice things about this is that if you look at this, we have an enormous amount of information from all the anatomic work and the literature on who these areas are and what they're probably doing, or at least the best guess of this. And using this, we can subset this and handle the massive multiple comparisons problem that you do face when you make 143,000 tests. So instead of looking at randomly in the connectome, we can look at a language case like this, where on the left side, you have a normal person, and you can see all the parcellations in the language system. And these on the y-axis is the same. And you can see it's mostly red because the language system is mostly firing it together. Even if you're not using it, it's keeping itself warm. It's kind of the concept of resting state. But on the right side, in a person with a glioblastoma destroying the arcuate fasciculus, which we happen to get in the scanner, you can see that you lose that correlation. There's a lot of blue. And while that's really cool to be able to see aphasia, it's more important that these are actually places in the brain. So that column is an area called 55B, which we've done a lot of work suggesting this is a unique motor cortex for the larynx. And it has a unique role in speech outside of Broca's area. And you can actually make that into an object and hit it. And that's kind of how we have a clinic that we do this pretty much every day. But utilizing things like machine learning, we can start to determine what's normal in that. So instead of just showing reds and blues, we show only things that are normal or abnormal. And what that means is that we actually have the ability to look at the connectome, the functional connectome, and determine what parts of the brain are asynchronous in ways that we don't like, and we can change them. We can stimulate them and change them. And to show you that this isn't, really in even the realm of something we need a randomized trial to say that at least this is worth exploring further. This is a patient who had a very large MCA stroke that we treated in our clinic. She's 18 months out from it. And you just have to trust me. This is a 3-dimensional coronal view that the slices in front of this, the entire superior division has been wiped out, and there's no arcuate fasciculus connecting Broca and Wernicke's area anymore. Despite this being a chronic deficit, we looked at it and said that we thought that her fundamental problem was that she needed the supplementary motor area of some kind to communicate to Broca's area better. And we thought that because her main problem was initiating speech. She actually had relatively normal speech when she could get things out. So we stimulated the contralateral SMA, and we actually repeated that image on the right 3 times because we had to go back 3 times because we didn't believe this. But essentially, you can see that she's actually begun to strengthen the connection to the other side. So in the interest of time, and I actually shut that off, I'll kind of wrap that up. But one of the thoughts that we're working on now is how do we utilize big data techniques to really begin to figure out where to look. And so what this basically involves is getting through all the challenges with hospital IT and infrastructure using smart techniques that are built for the hospital to allow us to really take advantage of cloud computing and machine learning at massive scale. So we have the ability now to run 10,000 scans through a processing thing in about an hour and run 30 or 40 different machine learning models to find which one has the best fit and look at different strategies. And so when you have that kind of scale, that lets you fail fast and be agile to try to find the best strategy for explaining the data. And what one of our thoughts is that as we begin to build this into the hospital architecture, we can begin to leverage the fact that we're generating massive amounts of data that never make it into a research study to really learn where do we look and to allow institutions to work across sites in ways that don't involve passing scans around or doing de-identification or all these other things that eat up large amounts of time and involve legal risk for data security. And hopefully over time, as we do that, we'll begin to understand where to look in the connectome and what things predict things that we care about. Thanks.
You know, Mike, I was going to introduce you with a joke, but I think it's more prudent now, which is that you are literally and figuratively coming to us from the future.
Yeah. That is in fact true. I'm on Saturday now, so
yeah,
it's great. It's
Saturday
great here.
morning, but I think you're about 10 years ahead of everybody else. Tremendous talk. I want to forward a question that was emailed directly to me from one of our neuropsychologists, and wondering about how you and your team are including baseline and ongoing neuropsychiatric evaluations and trying to use that data to help inform the connection models at an individual patient level.
Well, yeah, that could be an entire talk by itself. But in short, the biggest challenge is that the neuropsychological endpoints. First thing, you have to separate them out from each other. That's a pretty big statistical effort. And the second is that what you will ultimately do with machine learning is you say, this is the connectome, and this is what I want you to learn from it. Tell me what thing predict that. So every one of those neuropsychological tests is a target. And one of the things about that we've noticed with neuropsychology is that even if you have the best neuropsychologists in the world, they can't see everybody. I mean, no matter what, right? And they certainly can't see the GBM who is going into surgery and measuring. So we're not measuring our outcomes on. We have a lot of data on epilepsy patients because they have plenty of time to get ready for surgery. We don't have as much patients on the people who really have the cognitive morbidity. And so one of the things we thought about is, can we scale neuropsychologists through things like technology? And so we created an app that basically takes a lot of familiar neuropsych tests like modified versions of HVLT and things like this, and with the goal of keeping it under about an hour, because in our experience, it's about as much as most glioma patients will let you do or be able to handle, and utilizing things like machine learning to get an assessment of, from voice to text, can we detect aphasia? And the answer is about 92%. That's where we're at now. We need more things before I go and put hand on heart and take that to the FDA. But the bottom line is that's what we're performing at now. And so yeah, it's better to have a 5-hour neuropsych battery with a professional, but it's just we can't get that. I mean, and actually the person who pointed that out to me was Sean Grady, who ultimately said, I have some of the best neuropsychologists in the world, but they're at NIH giving talks. And so one of the thoughts is that if we could extend the neuropsychologist, we could get a much bigger data set and really focus the neuropsychologist on, looking at data at scale, improving the tests, et cetera. More importantly is if you do it electronically, you automatically can put it into the cloud and immediately have it ready for machine learning architecture. So we have an architecture that does that all automatically. So essentially it has the ability to know that Mr. 497 scans and Mr. 497's thing come from the same patient without actually knowing who Mr. 497 is. It does that with encryption keys. And so what that means is that you can basically link all the patient with their data without actually being able to have any possibility of identifying them, which is nice. And what that also means is that means nobody's entering spreadsheets into some computer that could get hacked or anything like that. And it really takes the scale and the speed at which we can make discovery like this to a tremendously different level in our experience, which is, we really can start to get the scale that we get at Amazon to real, and according to them, they don't really trust machine learning models that have less than 60,000 observations. Well, that's going to take us a long time at the rate we're going.